Hi, this is the first of two videos on the immune system, and this one will cover the innate immune system, and the second will cover the adaptive immune system. So I'm going to start with some definitions. Firstly, we have an antigen. An antigen is any substance which is capable of inducing a specific immune response. So this is more relevant to our adaptive immune system, as our cells will recognize an antigen and then produce a specific response. We also have PAMPs, PAMPs being pathogen-associated molecular patterns. Now, these are antigens that are very obviously different from our host, so they can be detected by the innate immune system very quickly as being different, and then that can be broken down using a more generalized response, and that's part of the innate immune system. We also have DAMPs, which are disease-associated molecular patterns and play a very similar role. So this is the key difference between the innate and the adaptive immune system. Our innate immune system is a very quick response to very obvious threats, whereas our adaptive immune system takes slightly longer to get going, but is able to recognize all of the threats, even those that are less obviously different, and then creates this immunological memory so that if the pathogen is to return, then you're still able to fight it off again and more quickly the second time round. We also have barrier immunity, which is sort of part of the innate immune system. So this is broken down into two parts. You have your physical barriers, which stop the pathogen from getting inside your body in the first place. For example, the skin or the mucus within your lungs catching pathogens. And you also have biochemical barriers. And these, for example, gastric acid, are more commonly for breaking down the molecules once they're inside your body, but before they're able to cause harm. So one of the key types of cell in the innate immune system are our phagocytes, and they're so called because they perform phagocytosis. So there are three types of cells which perform this. We have our neutrophils. Now these only play a role in the innate immune system, so they simply perform phagocytosis and nothing else. Whereas our monocytes and our macrophages are also able to present the antigens of the cells that they've phagocytosed, and that can help activate the adaptive immune system. So you can sort of think of that as once they've broken down the pathogen, they then pick up its flag and wave it to tell everyone that they've defeated it, and then the others know that that army is attacking them. So the process of phagocytosis involves recognizing a foreign antigen, and this is often going to be, as it's the innate immune system, our PAMPs and our DAMPs, and these will be engulfed by our neutrophils, monocytes, and macrophages. Now, this might be after complement or antibodies have performed what's called opsonization, and this essentially is when they mark something out as being something that needs to be removed. So that's part of the innate immune system, the adaptive immune system, but then the phagocytes are able to help with that because it marks even the more even the more complex pathogens, it marks them as being obviously different, so the phagocytes are able to play their role. So once they've engulfed this microbe, they then release their toxic chemicals into it, and that breaks it down from the inside. And then, as I said, our monocytes and our macrophages put the antigens on the outside of their cell to demonstrate the type of cell that they've broken down. So we also have natural killer cells. So all human cells in the body have something called MHC class 1, except for our red blood cells. Now, when you have an infection and cells become infected, or you have tumour cells, they will lose this MHC class 1 molecule. And our natural killer cells have two different types of receptor on their surface. They've got an activating receptor, which binds to an antigen, and then you also have the inhibitory receptor. And this normally will bind to MHC class 1, and if it sees that, it will recognize the cell as being part of its own body and will leave it alone. However, in these infected or malignant cells, it won't be able to bind to this MHC class 1. So you won't inhibit the action of the natural killer cell, and it will just be activated. And therefore, it will release these pore-forming molecules and then pump its own cytotoxic chemicals into the cell and break it down to kill it. So now we move on to talking about complement. And sorry this slide is a bit busy, but I'm going to try and talk you through it nice and slowly, and hopefully it will all make sense. So I'm going to start with the classical pathway, as there are three different ways in which complement can be activated. The classical pathway involves an antigen on the pathogen cell surface binding to an antibody, and this produces a complex, and it's called the C1 complex. This then breaks down into the C2 and C4 complexes. And C2 and C4 are then broken down further into C2A and C2B, and C4A and C4B. Now, C2A and C4B then come together, and they produce something called C3 convertase. And C3 convertase is an enzyme which has the role of breaking down our C3. And it again gets broken down into our C3A and C3B, and the C3B then has the role of breaking down C5 into C5A and C5B, and C5B, and then C6, C7, C8, and C9 all come together to form what's known as the membrane attack complex. And this is how complement breaks down other cells, and it causes them to swell up and burst because it's basically created all these little holes within the membrane of the cell. So next we'll talk about the lectin pathway, and this works in a very similar way. And you have something known as lectin within the blood, and then it's able to bind to something called mannose, which is present on the cell surface. And this then produces a lectin-mannose complex, and then this basically mimics the C1 complex, and the process continues in the same way as in the classical pathway. Our alternative pathway is a bit different, in that our C3 isn't an entirely stable molecule. So although the C3 convertase normally breaks it down, it's also able to sometimes randomly hydrolyze. And this produces our C3b and our C3a. Now, normally, you have enzymes which are able to come and break down the C3b, and so the process stops here. However, when you have a pathogen present, the C3b binds to the surface of this pathogen, and this protects it from the enzyme which usually breaks it down. The C3b is then able to perform its normal role of breaking down the C5 into C5a and C5b, and then the membrane attack complex is able to form. So, in essence, the pathogen, in protecting the C3b from being broken down by its usual enzyme, is the master of its own failure, because it then gets killed itself by what the C3b has produced. So, acute phase proteins. So, when we have cells in our immune system, they have receptors on their surface which are known as pattern recognition receptors. And these are the receptors that are designed for recognizing our PAMPs and our DAMPs. Now, these are what I talked about earlier, our pathogen-associated molecular patterns, which are the antigens that are very obviously different from our host cell. So when these PRRs get activated, they release our acute phase proteins. And the acute phase proteins are things like our C-reactive protein, procalcitonin. And that's why C-reactive protein is one of the things that you test for in the blood to see if you've got an infection present. You then have these these acute phase proteins binding to our protein, binding to our pathogen, and then these have protective effects for the body, so they can help to inhibit the pathogen and stop it from working as effectively and damaging other cells, or they can promote parts of the immune system, for example, the binding of antibodies and complement to the pathogen, allowing them to break it down more effectively. So this slide is designed to act as a summary of what happens in the immune system. So we have our pathogen-associated molecular patterns or disease-associated molecular patterns, and they're recognized by our antigen-presenting cell with its pattern recognition receptor. Now, this cell is able to both break down the cell itself, and it releases our acute phase proteins. So we've got our cytokines and inflammatory mediators. And these go on to cause inflammation, and then that's why you see inflammation in an infection. So these cytokines then go on to help recruit more of our phagocytes. So these are neutrophils, monocytes, and macrophages. Our neutrophils are obviously able to perform phagocytosis on the cells that they recognize as being non-self. And then our monocytes and our macrophages will do the same thing, but when they do it, they'll also present antigens to help activate our adaptive immune system. Now, one of the roles of our adaptive immune system will be to produce antibodies, and these will have their own effect of breaking down the pathogen, but they also have a role of activating the classical pathway of the complement cascade, and the complement will then form its membrane attack complex and break down our pathogen. We also have our alternative pathway, in which our pathogens essentially activate the complement system themselves, and again we have the breakdown by the membrane attack complex. Finally, I've included down here a natural killer cell and an infected cell, and at the top here we have our activatory receptor, and this is activated by an infected cell, and this is our inhibitory receptor, but because the infected cell doesn't have our MHC class 1, it's unable to inhibit the action of natural killer cells, so the natural killer cell will also break down our infected cell. And that's essentially how the innate immune system works. So the next video will be on the adaptive immune system. So I hope that was helpful. Thank you very much.